It's just a science project.
Silent breed is people!
You know, a doctor friend once said the same thing to me. Frankenstein was his name.
It's alive! It's alive! It's alive!
That sounds like something out of science fiction.
Please explain to me the scientific nature of the whammy.
We live in a spaceship, dear.
So? Yes, science!
Program complete. Enter when ready.
Hello and welcome to episode 216 of Science on Top. Today is Sunday, the 28th of February, 2016. I'm Ed Brown and I'm joined by Dr. Shane Joseph.
Hello.
Penny Dumsday.
Hello.
And Lucas Randall.
Hello.
And today we're going to be talking about the origins of fast radio bursts, a possible new cause of Lyme disease, and penguins that wobble. Well, it's true. But Lucas, fast radio bursts, FRBs, they're the sudden, very short blasts of high-energy radio waves. And at the end of last year, there was a paper that proposed they're the results of starquakes, these massive, Wikipedia refers to it as a sudden adjustment of the crust of a neutron star. And that may still be true, but a new paper published in Nature shows that a team of astronomers have traced the origins of an FRB to a galaxy 6 billion light years away. This is really cool.
Yeah, what's particularly cool about this is it's the first time they've managed to capture an FRB whilst it happened. All the other ones, the other 16 that we've detected, have been detected by going through the records. So in some cases they were found years after the actual event. So as a result, they never had a chance to sort of zone in on where they've actually come from. Radio telescopes don't have particularly high resolution, so they don't really give you the ability to zoom in and go, okay, well that's exactly where it's coming from in the sky. They kind of cover anything from like half a hemisphere to a few degrees of arc. So it's it doesn't give you the resolution you need to figure that out really, really quickly. But what happened this time? So go on.
I was just going to ask a pedantic question, just to clarify. This is something that came from a galaxy
far away.
Far away.
Right?
6 billion
Yes.
light years, yep.
Long ago.
Yeah. So actually the event happened 6 billion years ago.
Yeah.
Yes,
Yeah.
correct.
Yep.
Yeah, absolutely. So
That just blows my mind still. Anyway, yeah, continue.
I know, because the Earth's only been around for 6,000 years.
that's yes.
Ozing.
But still, no, even it's before the Earth was created,
Yeah,
4.3
it is,
billion
yeah.
years
Yep.
ago, before the solar system was around.
Yep. It was before the pill, before the dollar. Sorry, go on.
Wow.
Continue, Penny. Sorry.
No, it's just exposing my ignorance. I was like, I thought the solar system was 13 billion, but that's the universe, isn't it?
Universe, yes.
Mhm.
Yeah.
Yep. So as you said, Shane, it's 6 billion years ago when this event would have actually occurred, because it's taken that long for the light to reach us, give or take, because obviously there's expansion involved in that process as well, and that actually leads me to one of the things that was also really cool about this, because they were able to detect this as it was happening. They detected it through the Parkes radio telescope in Australia, of course, the one made famous in the movie The Dish. After detecting it, they were able to send out very rapid notifications to other astronomers around the world, who were able to then bring their instruments to bear, so to speak, and point them up in the sky to try and figure out where this was. So one of the other telescope arrays in Australia was brought onto about 2 hours later, and through that they managed to narrow it down to a particular area of the sky. That was the square kilometre array, and then they were able to get the one of the telescopes in Hawaii oriented to the signal as well, and further narrowed down where it's come from. And the cool thing was that although the fast radio burst itself only lasts milliseconds, so that was only detected by the Parkes radio telescope, there's like an afterglow or a series of pulses, if you like, that occur after one of these events, and that's what these other telescopes were able to detect to help them narrow down where it actually came from. Now, once they realized where it was from, they were then able to use the signal itself to measure how much material the signal had passed through between its host galaxy and reaching us. So traversing 6 billion light years worth of space to reach us, it's gone through a whole lot of stuff. And the different wavelengths actually travel at different speeds as they go through different mediums. So using this information, they're able to test one of the theories of the distribution of matter in the universe. It meant that they could, first of all, they could narrow down on what might have been the cause of this. And there's four main proposed causes for these fast radio bursts. One of them is pulsars, which are neutron stars that are spinning really, 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 really fast and giving off these pulses of light and magnetism. They're really cool, but they're basically, they don't stay in that state for very, very long. Then there's neutron stars that are collapsing, or perhaps neutron stars merging together, or disappearing black holes as they, finish their life cycle, if you like, because they do radiate energy out using Hawking radiation. They've even been suggestions that perhaps they're alien beacons, of course, because there's always aliens involved somewhere along the line.
Definitely that one.
Yes, 6 billion years ago.
So this particular observation was able to rule out pulsars because pulsars are the type of star that they are means that they don't last very long. And this particular galaxy is an elliptical galaxy about 6 billion light years away, which is quite old. And stars form in young galaxies. And
so how long did pulsars last for?
Oh, I couldn't tell you off the top of my head. They have an order of not very long in galactic terms, basically. So you wouldn't expect a pulsar to still be hanging around in a galaxy, an elliptical galaxy as old as this. Okay. So that meant it wasn't one of those. The leading theory at the moment is that this was actually a merger of neutron stars. Neutron stars are another type of star which were similar to pulsars. In fact, pulsars are a type of neutron star, but a neutron star is something that's finished its life as a main sequence star, and it's collapsed down into a really small sort of Earth-sized thing where the gravity is so strong that basically the matter all merges together in like a neutron mass, and it's just ridiculously heavy. They just basically sit there doing their thing. But the host star wasn't big enough to collapse all the way down into a black hole. So that's the first thing is figuring out, okay, this based on its signature, it looks like it would have been a neutron star. Interestingly enough, if the correct equipment was pointed at it, this may have been another opportunity to detect a gravitational wave, similar to the one that we talked about a couple of weeks ago, because this type of event would have caused a gravitational wave. However, don't have that observation. But the other thing, as I mentioned before, it enabled them to do was to use this signal to basically measure the amount of matter in the interstellar and intergalaxy space in between us and the host galaxy. And the universe, the scientists have known for quite some time, or proposed for quite some time, the universe is made of about 70% dark energy, roughly sort of 25-ish% dark matter, and about 5% the luminous matter, the stuff that we can actually see, the stars and so forth. But when they add it all up, there's still a lot of stuff that's not visible. We can't figure out where it is. And using this signal, they were able to dial in more or less on exactly how much intergalaxy or interstellar stuff it passed through, and it sort of lined up with the expectations, which is really, really cool. And it's the combination of knowing how far away it was, how long it had traveled, and the difference between the different wavelengths reaching us that enabled them to figure that out, which is really, really cool. Yeah. I won't go into all the how and all that sort of stuff, because there was one of the explainers which was really good. I think it was the Nature one. Which one was it, Ed? The
missing matter one was Universe Today.
Yeah, that was good, but it wasn't too detailed. Actually, that's a really good primer.
Science Alert was a bit vague.
Yeah, Science Alert was vague. Yeah,
so
but
there was been Nature.
I think it was the Nature one where there was quite a lot of background info talking about why this is important and so forth. But anyway, it was very, very cool, and the fact that it's the first time we've managed to do it, and it's really this incremental gains that we're having in instruments that's making such a big difference. In this case, it was the collaboration tools that made a big difference, because as the science team, the CSIRO science team that were monitoring this via Parkes, received notifications of this alert, they were able to forward it onto other astronomers instantly. And it means that other astronomers were able to make the phone calls and so forth that were needed to bring other instruments to bear, which meant they could capture the full story, because Parkes, couldn't see that other stuff. So
and also confirm it with other instruments.
Yeah, exactly. And that's really the key change here is that the team, this international team that was involved in this particular discovery, they had already spent time setting up this network in the past, because it was identified as a real problem, because they'd never managed to capture one of these signals. So the real key was being able to let all the other astronomers know early enough so they could bring their instruments to bear.
And in particular, getting the Subaru telescope in Hawaii onto it, because that's an optical telescope.
Yeah.
And that was able to then measure the redshift of the, I assume it was of the galaxy, not the burst itself, which wouldn't be invisible light.
Yeah, exactly.
And from that, they were able to calculate all that missing matter and so on. What do you know anything about that missing matter? Like, is it just that it was obscured by dust and other stuff? Why was it missing?
Yeah, pretty much. It's missing, but in terms of it is not known. Like, it's theorized that this is how much actual physical matter there is. This is how much dark matter there is, and so forth. And it's theorized how much is in that interstellar space, that interstellar medium. And it's very, very difficult to measure that, because you don't, like, in every direction around us, we're going to have differing densities of this interstellar dust and so forth. Figuring out how much of it is there means that you need something to compare it to. You need, like a baseline first and whatever. And the key, from my understanding of reading this story, my understanding was that the key here was the difference in the time that it took the different frequencies to meet, to reach us, because these frequencies are affected by, dust and little grains of different sizes. A little bit like, I guess, the Rayleigh scattering in the Earth's atmosphere causing the blue, sky. It's that's caused by a similar thing. It's wavelengths of light that are bounced around by different size particles, and that appears to be what the key was here. So, this is why it appealed to me, because there were three different sort of cool things about this story. It was the first time that it was a collaborative effort, that they managed to get the other, which led to them being able to get the other telescopes to bear onto it, and also the fact that it enabled them to to find some other very cool information, which was cool.
And also, I think that getting all the other equipment online is just an impressive feat when you consider this wasn't just the Subaru optical telescope. It was also a lot of radio telescopes and everything. These telescopes take a while to move around and get to the right position.
Well, yeah, and it actually struck me as being interesting as well, because a lot of these telescopes are tasked, months and in some cases years ahead as to what they're observing, and their observing runs can be, hours, days, weeks long, of whatever they're looking at. So it would have been quite a lot of negotiation involved with getting this project high enough on the agenda, I imagine, to say, okay, there's been one of these detections, drop what you're doing, shift over here and look for these frequencies.
Sorry, George, you'll have to reschedule that telescope time for another year away.
Yeah, exactly. And that's, I think when we had Pamela Gay on the show, we talked about this a little bit. It's just how devastating it can be when you've got your scheduled, observing run for months and months away, and you reach that time and they suddenly ditch you for something else. And it's goes, well, tough, rebook. You can get back in the queue again. It's, oh, no, my PhD relies on this data. So, yeah.
That's science, but that's limited resources science. So more funding, more telescopes, that'll fix it.
Yeah.
It's an interesting thing to say when the CSIRO was hugely involved in this discovery, and we're cutting funding to that and sacking people. And anyway, let's not dwell. Let's move on instead to penguins on treadmills. No, it's not a cuter, better-acted sequel to Snakes on a Plane. It's an experiment led by Astrid Williner from the University of London. And she set out to see how penguins walk, or waddle, how that changes with variations in body mass. And Penny, fat penguins wobble when they waddle.
They do indeed.
Which could also be a children's book.
This is one of those evolutionary payoff kind of stories. So what they found is by watching these penguins walk on treadmills, and when Ed links to the story, it's worth watching the footage. It's very cute. Is that the larger penguins are less stable walkers. So presumably, they're using more energy to walk, they're probably a little bit more vulnerable, and so on. They weren't able to test every penguin. Not every penguin could figure out how to walk on the treadmill, and some of them cheated. So they were sort of in a box on a treadmill, and they'd like lean up against the back of the box so they didn't have to walk. So
cheats.
That's pretty funny.
So anyway, because of the way that penguins rear their chicks, it's really important for them to put on a lot of weight because they need to sit and incubate that egg on their feet for extended periods of time. So I guess there's a point where they reach that sweet spot between not too fat to walk and fat enough to last a winter or last the incubation period.
I do find that actually, as much as I poo-pooed this story, just for the silly idea.
I know, Shane was very poo-pooing before.
But I am quite interested in this idea of the evolutionary trade-off, where it's basically that, as Penny said, that sweet spot between, in this case, I've got to be fat enough to take care of my child. At the same time, I've got to be able to get somewhere.
Yeah, and I mean, they don't have to get super far because once they're in water, the weight doesn't really matter once they swim. But I guess the other thing is, with climate change and the changing landscapes, like who knows how a penguin's travel is going to be affected and what that might mean for incubation. So I guess in and of itself, I was drawn to this story because I just found it very cute. But it does raise some bigger questions, as most of our stories do.
Yeah, and it's important to learn things like this and to figure out how energy expenditure is going to be related to their survival and how things like you don't think that, just eating and storing up all that fat is going to have an effect. But then there was also a story about 150,000 penguins that have died over the last 5 years in Antarctica when a giant iceberg kind of blocked off their route to the sea and they have to walk 60 kilometres to get to the nearest access point to the water and to feed. So there's all these reasons why you need to store up enough food to be able to survive for a long time, but you also need to be able to walk efficiently and then to swim well as well. And it's a really cool story.
It's cute. I would like to say, imagine these people at parties. Oh, what do you do? I'm a scientist. Oh, that's cool. What do you do? Oh, I watch videos of penguins walking on treadmills.
Well, yeah, it also, it's not an entirely great story because they actually had to kidnap these penguins, take them into a lab and study them for a bit on a treadmill. Yeah. Although it does say that when they weren't being studied, they were kept in pens close to the rest of the colony. So, all right, Lyme disease. Now, every year there are more than 30,000 new cases of Lyme disease in the US that are reported to the CDC, and even that number is probably underreported. It's the most common tick-borne disease of North America and Europe and one of the fastest-growing infectious diseases in the US. And for a long time now, we've thought it's caused by a single microbe, the spiral-shaped bacterium that Shane will now pronounce for us.
Now I'm under pressure. Borrelia burgdorferi.
Exactly.
Or B. burgdorferi.
But in a new study, pathologists from the Mayo Clinic, Dr. Bobby Prett, and her team found that Lyme could be caused, rarely, by a different bacterial species. And Shane, former Florida governor and wrestler Jesse the Body Ventura, once dedicated a whole episode of his show Conspiracy Theories with Jesse Ventura to Lyme disease.
Oh, did he really?
And it's a disease that is swamped in controversy. But this is an intriguing finding given the new species can cause more serious symptoms from vomiting to neurological issues.
Yeah. Look, I'm going to start this with the caveat that I know very, very little about Lyme disease. I'm a microbiologist by training, but I have never dealt with Lyme disease, obviously. I didn't even know there was a controversy about it. That's how little I know about Lyme disease. That's
okay. Jesse the Body Ventura is also not an expert. So,
well, he did his own research, Ed, just like you suggested.
That's right. Yes. He looked it up on Wikipedia.
So the interesting thing about the causative agent of Lyme disease is that it seems to be very, very hard to grow. Now, one of the main ways that we study diseases is you have to grow the organism from a sample. first of all, to prove that it's alive. And then I've mentioned Koch's postulates in the past, and I just got a silly snigger from Lucas every single time I did.
Well, I almost did it again.
You're going to do it again. But the point is that with these postulates is that you have to prove that this agent that you grew from the sample will then cause disease. Now, when it comes to something like Lyme disease and the causative agent Borrelia burgdorferi, it's actually very hard to grow in the first place. It's apparently just almost impossible to grow from samples. A bit like, I think, the syphilis causative agent as well. Just very, very hard to grow in the lab. So for this reason, I think this is another one of the controversies, I guess. It's just been very, very hard to study. So what these researchers did was they used a DNA technique instead to look at various samples that were given to them. This is in the Mayo Clinic. And I think they received samples from like a 10-year period. And they looked at these using DNA analysis, and they were looking for a certain kind of gene in these bacteria. And what they found was that if we look at the DNA sequences of the gene they were looking at, they're actually quite different to the type species B. burgdorferi. And in fact, they're probably different enough that they could be a new species. Now, that's all well and good. I'm not arguing that this is not a new organism. What I am arguing is that they don't actually know that this is the one that caused the disease, because they haven't proved the fact that this can then cause that disease. They haven't shown this in a clinical setting as far as I'm aware. Now, I could be wrong. Maybe I haven't read the paper in enough detail. But they haven't actually – there's no direct link here that this new species, which they're calling Borrelia mayoni after the institute, that this actually is the cause of the new Lyme disease, or a different Lyme disease, if you like.
Okay, so all they've really done is found that this bacterium is common in a lot of these samples. They haven't necessarily found a link between that and
as far as I'm aware,
Lyme disease
they haven't
symptoms.
proved that this is the one that's causing that disease. For all I know, I mean, the thing is, now, PCR is a funny beast. Sometimes, and I'm not saying this happened in this case, but sometimes in PCR you can, which is basically it's an amplify. What PCR does is it takes a sample, and you can amplify or create multiple copies of a certain gene you're looking for. In theory, this works great. And in theory, you can do this in a quantitative manner. But sometimes things go a bit funny, and especially if you're looking at genes that could be slightly different but not. And then sometimes your numbers can be all skewed. So I would like to actually read that original paper, but of course it's paywalled, as all these papers are. And I'd be interested to see what gene they use to identify these new species, because I'd like to see how different this was. I mean, as far as I'm aware, they're basing this entire well, no, actually, that's not true. They did grow B. mayoni. I'm wrong. They did actually grow this new organism. And funnily enough, this new species was actually very easy to grow, in contrast to burgdorferi, which is actually very, very hard to grow.
Okay.
But as far as I'm aware, they haven't actually, and this is, I think, where this feeds into this whole controversy thing. They haven't actually proved that this new species causes disease.
Yeah.
So, yeah.
And it's good that they're doing the research for it, though. And
definitely.
This is, as you say, there's so much controversy around there. There's also another researcher who was not involved in the study is, quote, troubled that the new paper did not mention any of the previous findings. And he says, I find it unprofessional and unethical to not give proper scientific credit to those other researchers.
Yeah.
And so there's just everywhere you turn when it comes to Lyme disease, there seems to be more controversy, more uncertainty, more debate, which is how science works, of course, and that's to be expected.
And again, I had no idea that in such a I wouldn't call it esoteric, but it's a fairly small avenue of research here. There is this much bad blood.
Yeah, I'll see if I can get someone on to shed more light and clarification on this.
And look, and if I'm wrong about them not proving B. mayoni is the actual causative strain of the causative agent of Lyme disease, please tell me, because as far as I'm aware, it's not. They haven't actually proved that definitively. I mean, they found it molecularly. And they found it. I mean, look, it's a bit of a smoking gun. They found it in samples of people who have Lyme disease, so it's quite it probably is the causative agent. See, this goes back in some ways to what I find interesting about the way we classify bacteria and different strains. Like the classical way of describing a genus and a species doesn't really work for bacteria, because bacteria can recombine DNA very, very easily as compared to, the so-called higher animals or the so-called higher plants. So imposing on them the same classification system that we have for, like the animal kingdom seems to be a little bit, it's limited. It's very, very limited. So it could be the case, and this is again coming from a total sort of a bystander's point of view, that you could have multiple strains that are recombining and all over the place and causing disease in different ways, and that's probably part of their strategy in some ways. It probably helps them to maintain this. So anyway,
I'm wondering, is it worth if we just quickly run through Koch's postulates, because I think it's pretty much the defining method of determining whether something is causative, isn't it?
Yes, it is. Now I've got to actually find the damn things.
I've got them here. So
Okay.
Koch's postulates basically are, number one, that the bacteria must be present in every case of the illness, although I think he's later retracted that because sometimes it can be present and not cause the problem, if that makes sense. They can be asymptomatic. Bacteria must be isolated from the host with the disease and grown in pure culture, and that's the difficulty that you have with the Lyme disease one. The specific disease must be reproduced when a pure culture of the bacteria is inoculated into a healthy, susceptible host. But again, some people can be asymptomatic to it, or you can have a pre-existing immunity. And the bacteria must be recoverable from the experimentally infected host.
Yeah. Now, they're the classical Koch's postulates, and there are obviously different ones that have been updated for the 20th and 21st century, where we had DNA techniques in which you can use a sequence of the DNA sequence of the pathogen to have as one level of proof.
Yeah.
So that feeds into that. But it essentially is still the same principle in that you need to be able to prove that this organism that you've obtained, or the DNA sequence of this organism, is actually the one that caused the disease in the first place.
Right, okay.
Yeah.
So yeah, that wasn't around in 1890
Obviously
when
not.
Robert Koch first put forth these postulates. But no, it's interesting to know how that sort of thing is done, and the ideas behind it all, I think, is fascinating. And do you want to touch on the bacterial nanospears and how are biologists stealing the nanospear technology from bacteria?
Yes. I love this story. this sort of I remember when I was I think I was still in undergrad actually, and the man who would later become my PhD supervisor told this story about an organism that used bacterial spears, like it was like a paramecium kind of thing, like a big ciliate, and it used these little, like basically projectiles that would, at the end of these projectiles were bacteria, and they would puncture cell membranes. Now this isn't the same system, but it's in some ways very similar. So paramecium, essentially they have like an almost symbiotic relationship with these bacteria called Caddybacter. And what Caddybacter do is they have these basically retractable spears that they express. And a certain paramecium species will release them in the, say, they're competing for resources with other paramecia genera, and other paramecium will envelop these bacteria as food, and then these bacteria will act as Trojan horses and produce these nanospears, or R bodies, which is what they've been coined as a bit more technically, refractile bodies. And these basically sort of coiled up proteins then will retract into spears and puncture the vacuole that they've been imprisoned in, get out, and in some way that they haven't figured out yet, kill their host. So,
right.
Yeah. And
that doesn't sound a very good thing to do in a symbiotic relationship.
No. Sorry, the bacteria the paramecium who had them release them as weapons.
Oh, okay.
And they get taken up by their competitors, and the competitors are the ones that are ruptured.
Okay, right. Sort of sabotage bacterium then. Okay.
Yeah. So this would have, if it hadn't gone any further than this, it would have been like a cool little sort of nature footnote. But this researcher has been like, well, why can't we use this to deliver genes and other things to other systems? So she's basically, this is Pamela Silver and I think, what's the other researcher called?
Polka?
Jessica Polka.
Yep.
Yeah. So they've essentially taken the genes from the Caddybacter that produce these R bodies, or nanospheres, if you want to call them that, and
I do.
Okay, fine, we'll call them nanospheres. Fine, child. She's inserted them into the gene of other well-known bacteria like E. coli and things like that, and she's figured out a system where she's basically figured out switches, so she can control the sort of the functionality of the R bodies that are then expressed. So if you lower the pH a certain amount, I think in this case, if you lower the pH of the system, these nanospheres will extend. If you then raise it up to a bit more of a neutral pH, they will retract. So that's just the starting point, and then from there you can imagine a whole lot of different ways you could go with this.
Okay. Yeah. So essentially, she's figured out how to make these bacteria poke out their spears and pull them back in. And the idea then is that we can maybe use that as a delivery mechanism for, genes, essentially.
Yeah, pretty much.
So like a viral vector to deliver a genome into an organism or
pretty much
targeted cell.
Yeah.
That is pretty cool.
That is pretty cool.
Awesome.
Yeah. I just like this. I love it when researchers sort of find these weird things that you wouldn't, you look at nature and go, oh, that's kind of odd, kind of cool, but odd. And then they think, oh, actually there could be an application for this.
Yeah, exactly. How can we use this? Yeah. And I just love Ed Yong's headline in The Atlantic, Biologists steal Nanospear technology from bacteria.
And Ed Yong also used the word apeshit in that article.
Which I thought was He's a colourful guy. All right. Was there anything else that we wanted to talk about?
Can I just correct something that I said earlier on? Just regarding pulsars. I mentioned the size of pulsars being around the size of Earth. I was actually thinking white dwarfs. Pulsars are much, much smaller than that.
They're like city size.
Yeah, they're like 10 miles or so across. They're really, really small.
But still have the same mass of a star.
Yeah, that's right. They might have a sort of a solar equivalent of one solar mass, but squashed into a tiny, tiny little area, and they would have started their life as a supermassive star that would have gone supernova.
So my next question was: so our sun, for example, won't become a pulsar?
No, our sun will very likely become a white dwarf. But the thing that's unique about pulsars is as a neutron star, they're very, very small, and that's why they're basically made exclusively of neutrons, because everything's squashed together so much. But what's really cool about pulsars is all of the rotational energy that the great big star had is compressed down to this tiny, tiny little point. So you end up with something that rotates with extraordinary velocity. So you have a class of pulsars known as millisecond pulsars, for example. They might be rotating on the order of thousands of times per second. That's how quickly they spin, and that's just because, just like that whole you would have heard before about the ballerina or the ice skater spinning and then bringing their arms in and it makes them spin faster. That's exactly what's happening. It's the rotational energy that the great big star had now in a much smaller point means it goes a lot faster, and that's basically a characteristic of pulsars. So sorry, I just wanted to add that just to clarify.
No, it's a cool thing. It's a good to clarify that.
Clarify, yes.
You weren't supposed to pick up on that, you bastard. All right, that's our show. So check out scienceontop.com/216 for links to all those stories. And also, if you want to get in touch with us or leave some feedback, you can also drop us an email, feedback@scienceontop.com, or catch us on social media. And probably one of the best ways you can help us out would be to leave a review on iTunes. That'll beef us up in the ratings and more people will get to hear us. So thank you, Lucas, Shane and Penny.
Thanks, Ed.
Thank you, Ed.
And a special thanks to Marcos Benamou, who's been our editor and is piecing together the schmozzle that we give him each week into something that actually resembles a show. So thanks for that, Marcos. And thank you, everyone, for listening. We'll be back again next week, putting science on top of the agenda. Join us then.
Like I said, this is the most existential crisis our civilization has ever known, and I wanted to speak out about that tonight because simultaneously, while doing this brilliant film that Alejandro directed, I've been doing a documentary about climate change. The time is now. It's imperative that we act, and I really wanted tonight. I feel so overwhelmed with with gratitude for what happened tonight. But I feel there is a ticking clock out there. There is a sense of urgency that we all must do something proactive about this issue. And certainly with this upcoming election, the truth is this: if you do not believe in climate change, you do not believe in modern science or empirical truths, and you will be on the wrong side of history. And we need to all join together and vote for leaders who care about the future of this civilization and the world as we know it.